Hello, this is Yashvin. In this video, I'll be solving paper 2 variant 1, October, November 2019, 9700 biology. Question 1. Figure 1.1 shows the structure of the amino acid glycine, which is the shortest or smallest amino acid with the shortest R group. A1. Name the parts of the amino acid molecule labeled A and B. So A, NH2 is the amine group and B is the carboxyl group. Two amino acids are monomers used to build proteins. Complete figure 1.2 by drawing a diagram to show the formation of a peptide bond between the two molecules of glycine. So what happens here is the OH from the carboxyl binds with either of the two hydrogen atoms of the amine group of the next amino acid. And so I'll include the lower hydrogen atom in this bond, which forms water. And then you've got a bond between carbon and the nitrogen of the next amino acid. This bond would be known as the peptide bond. And that is it. B plasma cells synthesize and secrete antibodies. Figure 1.3 is a transmission electron micrograph showing a plasma cell. 1 uses the label line and the label T on figure 1.3 to identify where the genes coding for the polypeptide chains of antibodies are located. So this gene would be a length of DNA present in the nucleus, and so you can draw a line to the nucleus and label that T. To calculate the actual diameter of the plasma cell shown by the line PQ, write the formula you use to make your calculation, show your working and give your answer to the nearest micrometer. The formula you'd use is you divide image size in micrometers with the magnification. The image size should be around 9 centimeters, and when you convert that to micrometers, that would be 90,000 micrometers divided by the magnification over here, which is 6,000, and that would be equal to 15 micrometers. 3. The plasma cell in 1.3 is very metabolically active. Suggest why there are very few mitochondria visible in the electron micrograph in figure 1.3. So a metabolically active cell is one that has a lot of chemical reactions going on inside. Reactions that need energy, which would be provided by mitochondria when it carries out aerobic respiration. So it makes sense that the cell should have more mitochondria than are visible in figure 1.3. And because this is a transmission electron micrograph, which was obtained using a transmission electron microscope, you'd obtain sections of the specimens of the cell's image, of the specimen's image, and so it is possible that the mitochondria of the cell are in the other sections of the specimen. C. Sieve tube elements in plants have very few organelles such as mitochondria. Explain how having very few organelles is an adaptation of the sieve tube element to its function. The fact that they have very few organelles means that there is less resistance to the flow in phloem sieve tube, and that means the rate of flow is faster. 2. Figure 2.1 is a transmission electron micrograph showing the bacterial pathogen that causes TB. A1. Name the pathogen shown in figure 2.1 that causes TB. So this could be Mycobacterium tuberculosis or Mycobacterium bovis. You can name any. The World Health Organization introduced a strategy in 2015 to end the global TB pandemic. An important part of the strategy is to identify people at risk of becoming infected with TB and using methods to prevent transmission of TB. The BCG vaccine is one method of prevention recommended for use in countries where TB is common. The BCG vaccine contains a non-pathogenic living form of microorganism that causes TB. 2. Complete table 2.1 by using a tick to identify the type of immunity that develops in a person who has been given the BCG vaccine. So a vaccine provides artificial active immunity. B. Rifampicin is one of the antibiotics used to treat TB. Rifampicin inhibits RNA polymerase in bacterial cells by binding to a site other than the active site. This prevents polypeptide synthesis. 1. Suggest and explain how rifampicin prevents polypeptide synthesis in bacterial cells. So rifampicin here acts as an enzyme inhibitor, and because it does not bind to the active site and rather to a site other than the active site, that means it changes the shape of the enzyme, which is RNA polymerase over here, such that it remains no longer complementary to its substrate. and so it prevents the binding between RNA polymerase and its substrate. As a result, mRNA is not synthesized, and if mRNA is not transcribed, that means translation in turn does not take place either, which therefore prevents polypeptide synthesis. Some bacteria have developed resistance to rifampicin. However, they are still susceptible to the other antibiotics that can be used to treat TB. Multi-drug resistant bacteria have developed resistance to at least 2 drugs, including rifampicin. WHO collected data from all of the countries on the number of cases of TB caused by rifampicin-resistant bacteria or RRTB and multiple drug-resistant bacteria or MDR-TB. Figure 2.2 shows the reported number of cases of TB between 2009 and 2013. So you've got a bar chart over here, and part 2 asks you to describe the trend shown by the data in figure 2.1. This is only of 2 marks, which means you can just state the general trend, which is increase in number of reported cases, and quote some data to explain how resistance to drugs such as rifampicin develops. I'll write 2 parts to this answer. The first one would include how resistance arises in the first place, and in the second one I'll describe how it can spread. So starting off, resistance arises by mutation, which can be due to overuse of the antibiotic or the course not being completed entirely. Now this mutation occurs in a gene of the bacteria. And because rifampicin binds with enzyme, resistance to rifampicin would mean that the shape of the enzyme changes and it prevents binding. And the second part would include that the resistance can spread either by vertical or by horizontal transmission, where vertical transmission would spread the gene for resistance within the same species. So when the bacteria divides into 2 by binary fission, or by horizontal transmission, where the gene is passed on to a bacterium through a plasmid. 3. A student carried out an experiment to investigate the effect of increasing the concentration of sucrose solution on the mass of potato tissue. So these potato tissue were cut into cylinders of equal length and diameter. The mass of each cylinder was recorded, and each cylinder was put into a sucrose solution as shown. After 1 hour, each cylinder was removed, blotted dry, and reweighed. The percentage change in mass of each cylinder was calculated, and the experiment was repeated thrice. The mean percentage change in mass of the potato cylinders in each sucrose solution was calculated, and figure 3.2 shows the results of this investigation. Alright, first off, on the x-axis you have got the concentration of sucrose, and the y-axis indicates the mean percentage change in mass of the potato cylinder. So up to 0.2 mole per dm cube of sucrose concentration, the mean percentage change is positive, indicating that mass of potato cylinder increases, and beyond that the mass decreases as the percentage change is negative. A, with reference to figure 3.2, describe the effect of increasing the concentration of sucrose solution on the mass of potato cylinders. So as I mentioned earlier, you would describe how up to 0.2% the mass of potato increases, and then it decreases beyond 0.2. And at 0.2 mole per dm cube of sucrose concentration, there is no change in mass because concentration of sucrose is equal to the concentration of potato cylinders. So there is no concentration gradient, and so the water does not move. And that leads me to my next point, that change in mass is due to movement of water either into or out of the potato. So when the concentration of sucrose solution increases, that means its water potential decreases. So now the water potential of a sucrose solution is lower than the water potential of the potato, and so the water would move out of potato into the sucrose solution, which would lead to decrease in mass of potato cylinder. And that's all you need to write in your answer. B. Explain why there was a change in mass of potato cylinders in 0.6 mole per dm cube sucrose solutions. They're asking for the reason as to why mass of potato cylinder changes, and at 0.6 mole per dm cube sucrose solution, the sucrose solution has a greater concentration or a lower water potential than the water potential of the potato cylinders, which would mean water moves out of the potato into the sucrose solution and down the water potential gradient by osmosis, and therefore its mass decreases. 4a. Figure 4.1 is a photomicrograph of a human blood smear. Name the cells labeled A and B in figure 4.1. Since A is larger than B and has a lobed nucleus, it's going to be a neutrophil, and B, being the smaller one, would be the lymphocyte. And this one over here is a monocyte. B. Blood and lymph are both fluids that transport substances within the human body. Table 4.1 shows the components found in both blood and lymph. Complete table 4.1 to show whether the concentration of each of these components is higher or lower or the same in the lymph when compared with the concentration in the blood and iota. You may use the words higher or lower or same once, more than once, or not at all. Oxygen's concentration in lymph is lower since blood in iota has just been oxygenated. The blood plasma that drains to form tissue fluid and then drains into lymph has passed through a lot of cells which have uptaken oxygen, and therefore they have a lower concentration of oxygen, and that is the same reason that they have a higher concentration of carbon dioxide. And lymph also has a lower concentration of red blood cells since it's not easy for them to leak out of capillaries due to their size. 2. The presence of a pathogen infecting the body leads to an increase in the concentration of protein in lymph. Suggest an explanation for this increase in protein concentration. Infection due to a pathogen stimulates an immune response, which in turn could increase the concentration of antibody, which is a protein. C. Blood is circulated around the body by the heart. The action of the heart is coordinated and controlled by the structures located in its walls, such as sinoatrial node and the atrioventricular node. One described the role of sinoatrial node. So it acts as the pacemaker by sending out waves of excitation that initiate the cardiac cycle. 2. In a healthy heart, the atrioventricular node provides the only pathway for electrical impulses to travel from the atria to the ventricles. The bundle of Kent is a structure found in the heart in a small number of people. Some electrical impulses do not pass through the atrioventricular node but travel directly from the atria to the ventricles through the bundle of Kent. Suggestion explain the effects that the presence of the bundle of Kent may have on the heart rate. The atrioventricular node not only relays the impulse as it travels from atria to ventricles, but it also delays the impulse, meaning that by passing through the bundle of Kent, the impulse won't be delayed. This delay by atrioventricular node allows the atria to contract first and then the ventricles after they have been filled with blood. Due to the bundle of Kent, the heart rate would be faster, as asked by the question, as the impulse is not delayed. That means there's a little time difference between contraction of atria and ventricles, and as a consequence, ventricles would contract way before they are filled with blood. 5. Myasthenia gravis is a condition that results in muscle weakness by affecting the immune response. A. Explain what is meant by the term immune response. So here I'll write down the definition of immune response, which is a series of reactions in response to foreign antigens involving the activity of phagocytes and lymphocytes. B. Myasthenia gravis is an autoimmune disease which disrupts a cell signaling pathway involving muscle cells. 1. Suggest how the immune system acts to disrupt this cell signaling pathway. In myasthenia gravis, T helper cells are specific to some receptors present on muscle fibers, form neurotransmitter that would stimulate contraction. And these T cells stimulate B lymphocytes to produce antibodies against these receptors, so that these antibodies bind with the receptors and prevent neurotransmitter binding with the receptor, and therefore these antibodies block impulse transmission. If impulse is not transmitted, that means muscle contraction does not occur. Enzyme Y is found on the cell surface membrane of muscle cells Enzyme Y acts to break down the cell signaling molecules which trigger contraction of muscles when they are no longer needed. 2. Using the induced with hypothesis of enzyme action, explain how enzyme Y breaks down the cell signaling molecules. Alright, since this question is of 4 marks, I'll write 1 point that is general to all kinds of enzyme is that they lower activation energy. And as for being specific to enzyme Y, which uses induced with hypothesis, I state how the enzyme's active site, or in this case enzyme Y's active site, is not complementary to the substrate, which is the cell signaling molecule over here. And so the active site changes its shape to mold around and fit with the signaling molecules to form enzyme substrate complex. C. Rituximab is a monoclonal antibody used in the treatment of myasthenia gravis. Rituximab acts against the cell surface membrane protein CD20. This protein is found in the surface of B lymphocytes that cause myasthenia gravis. Explain the advantages of using monoclonal antibodies in the treatment of diseases such as myasthenia gravis. In this answer, usually I'd use advantages that can be used for regular antibodies as well. And so it's that monoclonal antibodies are specific to particular antigens, which is also true for a regular antibody. And so they bind to that particular antigen, which in this case is CD20. And then you'd have to describe how an antibody leads to destruction of this particular antigen. And I'll be writing how it attracts phagocytes to carry out phagocytosis. 6a, outline the role of DNA polymerase in the replication of DNA. So this enzyme forms phosphodiester bonds between nucleotides in the new DNA strand. So this joins the nucleotides together, and it only adds nucleotides to the growing strand if they have bases that are complementary to the bases on the old DNA strand, which is a feature of DNA polymerase you should be well aware of. Floxuridine is a drug used to treat cancer. 1. Floxuridine inhibits DNA replication. Suggest why the inhibition of DNA replication is an effective treatment to kill cancer. So cancer cells divide uncontrolled, and if DNA replication does not occur, that means mitosis is prevented also, which is essential for cancer cells. 2. Floxuridine prevents thymine nucleotides being produced, and explain why floxuridine does not affect transcription. That is because transcription only uses RNA nucleotides and thymine is not one. It is a DNA nucleotide. It uses uracil instead of thymine. C. Figure 6.1 is a scanning electron micrograph of a lung cell in the stage of the cell cycle. 1. Name the stage of the cell cycle shown occurring in figure 6.1. The division stage of the cell cycle is known as cytokinesis. 2. Explain why smoking tobacco increases the risk of developing lung cancer. In this answer, you need to link lung cancer with the component of tobacco smoke that causes it, which is carcinogens. So tobacco smoke contains carcinogens that lead to mutation, and due to mutation, tumor suppressor genes are switched off, and this leads to formation of tumor or cancer. And so what happens is the cells divide uncontrolled. Alright, this is it. We are done with this paper. Thank you for watching.